Hello and welcome to today's podcast, where we're considering the evolution of oral biologic delivery. The physiology of the human gastrointestinal tract, or GIT, has evolved over thousands of years to create a luminal environment designed to host the microbiota needed to aid digestion of raw pre-agricultural foods and absorb available nutrients. The advent of cooking, the transition from hunter-gatherer to agrarian societies, and exposure to new pathogens resulted in the human GIT evolving distinct morphological, metabolic, and immunological features, adapting it to optimize nutrient absorption and creating barriers to harmful toxins and pathogens. Today, the rapid recent evolution of our diet and increased consumption of semi-processed and ultra-processed foods appears to have overtaken our gut evolution. This has disrupted the delicate balance of our gut microbiome, which is now being associated with increased prevalence of many diseases affecting digestive, immune, and neurological function, potentially creating a need for biologic therapies that can be delivered intact to rebalance the gut microbiome. Over the course of evolution, human GIT epithelia have also evolved sophisticated barrier and immune functions, designed for the selective uptake of valuable nutrients and to protect us from potential toxins and pathogens. Additionally, the human GIT contains a barrage of secretions from acid to enzymes designed to destroy pathogens and help break down foods into absorbable, bioavailable nutrients. These physiological features now present oral formulators with significant challenges in delivering complex biologic therapies intact by oral administration and achieving clinically viable levels of localized and systemic bioavailability. I'm joined today by Dr. HDR Vincent Jannin, currently Director of R&D and Head of Innovaform Accelerator at Lonza Capsugel, to discuss how oral delivery systems are evolving to overcome these challenges and the future prospects for clinically viable oral biologic drug delivery. With over 2 decades experience of leading pharmaceutical research programs and previously a lecturer in formulation sciences at Lyon University, Vincent holds a PharmD and PhD in pharmaceutical technologies from the University of Bourgogne and an HDR from the University of Lyon, recognizing his expertise in advanced drug delivery systems and making him ideally placed to help us explore this topic. Vincent, thank you for joining us and welcome to today's podcast.
Thank you, Andrew, for the invitation today.
It's our pleasure. It's always great to have an expert on a podcast who can hopefully answer some questions we'd like to put to you today. So with the increasing focus on biologic APIs, is the human GIT a viable route for delivery and uptake, or are we just trying to defeat gut evolution?
I believe that the gastrointestinal tract remains a highly viable route for delivering complex biologics if we design solutions that work with its physiology rather than against it. Our improved understanding of GI tract dynamics has already enabled strategies like leveraging pH gradients along the tract to optimize the release, solubilization, and absorption of small molecules. Nature also provides great inspiration. Lipids, for example, protect pancreatic lipases, which are proteases, from degradation by proteases in the duodenum. And similar principles can be applied to safeguard sensitive molecules. That said, there are 4 hurdles to achieving clinically meaningful oral bioavailability: stomach acid, gut enzyme, microbial action, and permeation across the mucus layer and intestinal epithelium. These are significant challenges, but insurmountable, and with innovative formulation and delivery technologies, we can defeat this obstacle. The reason it's worth pursuing is clear also. Oral dosage form offer unmatched advantages: patient acceptance and simplify logistics, potential for both systemic and local effect, which is relevant specifically for disease like Crohn's disease or colon cancer, where we need to have a target release of the drug for local action, and also broader access, especially for patients with needle fear. We've seen the commercial impact of the oral delivery of biologics already. For example, with Ribelsus semaglutide tablet, this new drug product drove a 14% revenue increase over Ozempic, the injection modality, alone in 2023. So rather than trying to defeat gut evolution, the future lies in harnessing its physiology while innovating to overcome this barrier.
It's interesting you mentioned that example of the increased revenue with Ribulus. And is Wegovy also now into a situation where they have a pill form, a tablet form, and there's, I believe, a oligoliform, I forget the exact name, I'm sure you know it, is also in clinical trial with Eli Lilly as well. So there's clearly a lot of focus, particularly on the GLP-1 agonist class of biologics. It's obviously a proven case. So how hostile is the stomach to biologics, and how effectively can complex acid labile organic molecules be protected from stomach acid?
So the stomach is indeed a very hostile environment for biologics. Its low pH, combined with proteolytic enzymes like pepsin, create significant challenges for acid labile molecules, like, for example, peptides or proteins. Protecting these molecules requires robust strategies that go beyond conventional enteric coatings. At Lonza Capsugel, we've developed a breakthrough technology, the Capsugel and Protect capsule, the first bi-layer capsule on the market designed specifically for enteric delivery. So since it is a bi-layer capsule, it features an inner layer of hypromellose, HPMC, a polymer known for its mechanical strength and compatibility with diverse fill materials, so it will be in contact with the formulation. And on the outer layer, we have hypromellose acetate succinate, HPMC-AS, an enteric polymer that will dissolve only at pH above 6.0, and it will be in contact with the intestinal lumen. So this design ensures the capsule remains intact in the stomach and release its content reliably in the jejunum or ileum. Clinical studies in healthy volunteers under both fasted and fed conditions have demonstrated consistent performance, delivering robust release regardless of dosing condition. Importantly, this capsule is compatible with high-speed feeding machines thanks to its coni-snap design, making it scalable for commercial production. Beyond acid protection, Protect capsule has proven its ability to safeguard biologics like protein, and we had an example with pancrealipase, delivering equivalent activity in the gut compared to marketed reference, using only the capsule shell for protection. It also protects peptide and microbiome-based therapies. We have shown that with fecal microbiota transplant application, the FMT, where the technology preserves microbiota integrity during processing and storage. This delayed release innovation represents a significant step forward in making oral biologics and microbiome therapy clinically viable.
So it sounds like there's a very robust solution in terms of acid protection. What challenges to gut enzymes and proteolytic action present for oral biologic delivery, and how can they be overcome?
So gut enzyme and proteolytic action present one of the biggest challenges for oral biologics delivery. These new drug modalities like the peptides, and we will talk about the GLP-1 agonists, but also the peptide also, the proteins, the nucleotide are inherently fragile and highly susceptible to degradation in the upper gastrointestinal tract. In the stomach and duodenum, they encounter a cocktail of digestive enzymes, proteases, amylases, lipases, RNases, all designed by an evolution to break down complex molecules efficiently. To overcome this, we need multi-layer strategies. First, physical barrier, like a delayed release capsule, can shield the payload until it reaches a safer environment. So we've discussed how capsule shell, such as those in the Capsugel and Protect capsule technology, provide a robust protection against proteolytic enzymes without requiring complex formulation. Second, formulation approaches in the capsule can add another layer of defense. Lipid-based systems are particularly promising because the proteolytic enzymes are not soluble in the lipid, so they cannot enter the lipid droplet where the API lies. So this creates a protective microenvironment for sensitive molecules, reducing enzymatic degradation and improving their stability.
And of course, they also potentially provide the keys to unlocking both intra and intercellular uptake as well in that process. I was taught a long time ago that the lipids are the keys to the human body. So how do gut microbiota impact bioavailability, and is oral delivery viable for microbiome therapies?
So this is correct that gut microbiota play a dual role in oral delivery. They can impact drug bioavailability and stability, but they also create opportunities for targeted therapies. For microbiome-based treatment like FMT, oral delivery is absolutely viable and transformative for patient experience. Compared to standard of care, to take just a capsule or several capsules, it's much better. At Lonza Capsugel, we've enabled this with Capsugel and Protect capsules, which are used clinically to administer FMT. These capsules have proven compatibility and stability with both the lyophilized solid formulation of FMT and the liquid glycerol-based FMT formulation. In collaboration with Paris Hospitals, we have validated enteric performance even after 2 years of storage at minus 80 degrees C, which is standard for these products. The capsule maintained integrity and performance under this condition, ensuring reliability for clinical use. Beyond microbiome therapies, gut microbiota can influence drug metabolism. Pharmaceutical industry has leveraged this advantage in prodrug strategies already. For example, sulfasalazine is cleaved by colony bacteria into an active 5-ASA for treating inflamed bowel disease where it is needed. Similarly, biodegradable polymers can be added into capsule shell to release the payload as a response to the enzyme from the microbiota and answer a local delivery very precise where we need into the games.
And I think this section is very important because, as I mentioned at the outset, there's increasing evidence, of course, that the gut microbiome, the so-called gut-brain axis, even in terms of neurological conditions, is a very important area of research right now. And I think there'll be a growing need for the ability to provide APIs that, or not APIs necessarily, but of course therapies that actually rebalance the gut microbiome. In what ways does the site of release influence bioavailability? Is it influential?
Yes, the site of release can significantly influence bioavailability, especially for drugs with a defined absorption window. For most small molecules, absorption occurs primarily in the upper GIT, so early release in the intestinal lumen can extend the residence time and improve uptake, particularly for drugs with low permeability. But local delivery is equally critical for therapies that act at specific sites. For example, pancreatin enzymes needed for enzyme replacement therapy have to be released in the upper intestine where they need to digest the food. At the opposite side of the gut, the anti-inflammatory drugs to treat Crohn's disease need to be released in the terminal ileum or in the colon to treat the inflammation. For new modalities, the picture changes. Avoiding the upper gastrointestinal tract can actually be an advantage because that's where most of the degradation occurs due to the acid and the enzyme. In these cases, delayed release becomes essential. Also, some peptides have demonstrated higher permeability in the lower intestine than in the upper part, and sometimes even higher absorption in the colon section. So with our bilayer capsule technology, we can precisely control the site of release. Capsugel and Protect capsule releases in the ileum, and we've developed a new version using a different enteric polymer grade that achieve an earlier release where we can target the jejunum, and we have proven that in human studies.
So it can be quite an important factor in achieving that level of clinical bioavailability that is necessary to make oral delivery a viable option for people. Are these new formulation strategies improving absorption and uptake and bioavailability?
So new formulation strategies are making a real difference in improving absorption, uptake, and viability, especially for challenging modalities like the peptides or the protacs, which suffer from poor permeability across the stomach and intestinal barriers. One promising approach, again, is lipid-based formulation. It can be combined for peptides, for example, with techniques like hydrophobic ion pairing to increase the drug lipophilicity and increase the solubility of the drug into the lipid system. For protacs, which are generally lipophilic, it is not needed. So these lipid-based formulation can form very fine colloids in contact with the intestinal lumen with particles below 100 nanometer, and these very small colloids can diffuse very efficiently through the mucous barrier. Going through the mucous layers, they come then into contact with the intestinal epithelium, and lipid-based excipient again can contribute also to improve absorption, improve permeability. For example, medium-chain fatty acids like C10, the sodium caprate, for example, can transiently open tight junction between epithelial cells to favor the paracellular uptake. Other lipids, like the long-chain unsaturated lipids like oleic acid, can fluidize the cell membrane and favor the transport of the drug through the cell. These long-chain lipids can also promote the secretion of chylomicron and promote lymphatic transport of very highly lipophilic APIs, bypassing the first-pass metabolism. So with the lipid-based system, we have a cocktail of molecules, lipids, that can act as permeation enhancer by themselves, like some example, the caprylo-caproyl polyoxyl glyceride, an excipient that is known to open tight junction, or other lipids, like vegetable oils, can generate in situ some fatty acid to open tight junction due to the cleavage of the ester bonds between the glycerol and the fatty acid, and release locally the fatty acid for this effect.
So from what you're saying, actually, a lot of the strategies are working with the physiology of the human body more so than trying to overcome the barriers as such. Do you think this has affected thinking generally on how to formulate for oral dosage forms?
Yes, absolutely. The thinking on formulation strategies has evolved significantly. We've moved from viewing oral dosage form as passive carriers to designing active delivery systems that combine multiple technologies to work with the physiology of the GI tract rather than against it. One example is, again, if I stick to lipid-based formulation, we tend to mimic natural processes. So just as the gut uses bilipid to protect pancreatic enzyme from proteolytic degradation, we can use lipid formulation to shield peptides and protein. Similarly, lipid nanoparticles, the LNPs, and the liposome that were originally developed for injectable modalities like the COVID vaccine are now being explored for oral delivery of mRNA and proteins through the gut. So this system mimics cell membrane with a lot of lipids in the cell membrane. And these lipids can enhance solubility, protect the payload, and also we use some specific lipid, some ionizable lipids that will facilitate endosomal escape to release the drug in the cytosol. Also, by adjusting the lipid composition and structure, for example, if we have a longer fatty acid chain with a higher melting point, we can even modulate the rate of API release that will enable sustained therapeutic effects. So this is a clear shift toward leveraging biology and material science together to create smarter, more adaptive delivery systems.
Yeah, it's very clever stuff. And as you say, I guess this ability has evolved as we better understand gut physiology, which is very complex in any event. So how does this affect the evolution of capsule technology? Do capsules have to change to support this approach to active delivery?
Yes, you're correct. So capsule technology is evolving rapidly to support these new formulation strategies and meet the challenges of oral delivery of complex molecules. So advanced polymer formulation have expanded the capabilities of capsule, enabling improved viability and stability for sensitive APIs. We recently published a review paper on emerging capsule technologies for targeted oral drug delivery, highlighting several key innovations. One major advancement is bilayer capsule, which allow us to combine different material to achieve specific target release profile or improve capsule shell formulation compatibility. So I already gave the example of the change of the enteric polymer when we can select specific HPMCS grade to target specific region into the intestine, like the jejunum or the ileum. But we can also modify the inner layer, the layer in contact with the formulation, to be sure that it will be compatible with the fill material, for example, with some lipid excipient or permeation enhancer. So this bilayer capsule can be customized on both layers, offering flexibility for diverse modalities. Beyond material innovation, manufacturing technologies are transforming design possibilities. Additive manufacturing, like 3D printing, enables intricate structure and precise control over drug release by spatially placing enteric materials layer by layer. Taking this further, 4D printing introduces stimuli responsive materials that adapt over time or in response to an environmental triggers like pH, temperature, or enzyme activity, creating dynamic, intelligent delivery system. Also, capsules are being integrated with intragut injection device. So, for example, we have the BioNDND from BioGrail for gastric delivery, and on the other side of the gut, we have the Ranipig from Rani Therapeutics for intestinal injection. And this intragut injection device enable achieving bioavailability levels far beyond standard permeation enhancer formulation for peptides or other fragile molecules. So capsule technology is no longer just a carrier. It becomes a platform for innovation by combining advanced material, smart design, and novel delivery mechanisms. We can transform capsule into active enablers of the next generation of all therapeutics, redefining what's possible for patient care worldwide.
Some of the technologies you've spoken about there also, of course, create the opportunity for combination therapies, and it opens up a whole new range of possibilities, I guess, in terms of therapeutic delivery. So right now, which classes of biologics do you think show the most promise for oral delivery or even potentially viable today? Obviously, we've spoken about the GLP-1 agonist class, which seems to be gathering momentum very rapidly. What do you think will be next?
So today, the most viable classes of biologics for oral delivery includes, of course, hormones, enzyme replacement therapies, and microbiome-targeted treatment like FMT, the fecal microbiota transplant. These have already demonstrated clinical feasibility and patient benefit when delivered orally. Looking ahead to the next 5 years, we see exciting progress for peptides, especially for the GLP-1 agonists, but also for protein, RNA-based therapies, and PROTACs. These modalities are challenging due to the size, fragility, and permeability issues, but advances in formulation science, capsule technology, and enabling technique excipients are making them increasingly realistic for oral delivery. And the promise here is transformative. Moving from injection to oral therapy for complex biologics will redefine patient experience, improving adherence and expand global access. So by continuing to innovate at the intersection of biology and material science, we can turn what was once considered impossible to the next standard of care.
And now I'm going to put you on the spot. How do you see oral biologic delivery evolving in the future? What's just over the horizon?
So, good question. So first, I see oral biologics evolving dramatically over the next decade. So in the short term, like I explained, it will be mostly peptide, protein, and microbiome therapies. But looking further ahead, over 10 to 15 years horizon, I believe we will see clinically viable oral delivery of monoclonal antibodies. Achieving this will require breakthroughs in formulation science, capsule technology, and enabling excipient or in situ injection devices, combined with a deep understanding of the GI tract physiology. What's exciting is that the evolution is just about overcoming barriers. It's about rethinking oral dosage forms, active delivery systems that work with biology rather than against it. We've already seen this shift with lipid-based formulation, bilayer capsule, and even emerging technologies like 3 or 4D printing for precision release. And this innovation will enable site-specific delivery, protection against enzymatic degradation, and improve permeability, all while maintaining patient convenience and global accessibility. So the future of oral delivery isn't just incremental, it's transformative, and it's within reach.
Fascinating. Thank you so much, Vincent. It really does sound like exciting times in oral drug delivery with new formulation technologies and advances in capsule technology already making successful oral formulation of biologics a reality. It also sounds like the prospects for more subclasses of biologics to be delivered viably in this way are also looking very positive, certainly within the time horizons of a drug clinical development cycle. I particularly like the idea that delivery systems are being designed to work with gut physiology rather than against it, and working in an active way rather than in a passive way, in delivering their role as in achieving clinically viable levels of bioavailability. From what you said, it seems like Lonza Capsugel have already gone a long way towards overcoming many of the physiological barriers to oral biologic delivery, particularly those presented by the human gastrointestinal tract. It's also clear from our conversation that Lonza Capsugel are committed to continuing to pioneer new oral dosage innovations and technologies. For example, your recent investment in Tampa, the CDMO site, will give you access to even more scientific and formulation expertise, and an ability, of course, to work with pharmaceutical partners from first formulation to rollout and commercialization. You really become an end-to-end oral delivery solution provider in many ways. Now that clinically viable levels of bioavailability have been achieved for oral biologics, it seems the potential for growth of this mode of administration is enormous, creating both possibilities for new oral forms for new drugs, but also, I guess, the chance to revisit existing drugs and reformulate in an oral form. Thank you, Vincent, again for a fascinating discussion and your insights into where we are today and what the future might hold, which certainly sounds very positive. If you would like to learn more about how Lonza Capsugel could support you in achieving a viable oral dosage form for your biologic API, simply scan the barcode on the screen. Thank you for listening today, and look out for the next podcast from Lonza Capsugel, where we'll be exploring how oral dosage forms can increase the commercial and therapeutic value of biologic therapies. Thank you, Vincent, and goodbye to our listeners.